All right, thanks everyone for coming to this week's seminar. We're really excited to have Nancy Emery here today. She's an associate professor at the University of Colorado. She's a plant evolutionary ecologist that studies evolution of habitat specialization in plants. So she did her PhD at the university at UC Davis in California. And then she moved on to Berkeley, also in California, before going to Purdue University. So she's traveling around Europe at the moment, and we're excited that she decided that Seville would be one of her stops. So yeah, take it away.
Thank you so much for being here and for letting me speak in English. You don't want to hear my Spanish. It's just really exciting to be here for many reasons. First, this is the first talk I've given in person in 2 years, I think, so I might get tired. We'll see. I'm very excited to be in Sevilla. I wasn't sure the pandemic was going to allow it to happen, and I'm very excited that there was a slot open that I could kind of drop in and give a talk on my research. And so Christoph has been an incredible impromptu host. I just invited myself for the most part, and he's made that possible. I am on sabbatical this semester, and for this first half of my sabbatical, I primarily have to work on writing projects, but I didn't want to miss the opportunity, if the pandemic allowed it, to potentially see some other parts of the world that I've always wanted to see, and go to places where, if the pandemic would allow it, I could drop in on amazing groups like this. And so several colleagues of mine, Dan Doak, Greg Gilbert, and Ingrid Parker, who I understand did a full year sabbatical here a few years ago, all said I had to try to come and say hello and introduce myself to you. So thank you for making that possible. I'm going to tell you the same thing that Christoph just did, but with a map, in case you're not familiar with my geography. So I actually did my undergraduate work at Brown University, where I studied salt marshes, and that's where I first got interested in ecology and evolutionary biology. And then I jumped to the other coast, where I did my PhD at UC Davis and my postdoc at Berkeley, which is very close by. This is where I fell in love with plants that grow in Mediterranean climates, which is part of the reason I'm excited to be here. In another Mediterranean climate, it feels very familiar. And then I had a position, an assistant professor position at Purdue University. This is near Chicago in Indiana, for about 5 years. And then was able to, I'm kind of spiraling in here, aren't I? I finally got the position that I have now at the University of Colorado, Boulder. So Boulder is in an amazing place if you like to study environmental gradients like I do. We say it goes from grasslands to glaciers within a very short distance. And so the campus, which is actually right here, lies right at the base of the southern Rocky Mountain Range. We call it the Front Range because it just comes right up and then stops, and then it's the Great Plains right after that. It's a beautiful campus right here in these, we call these the Flatirons. Really would love anyone to come visit. I'd love to help make that possible if you can. And at Boulder, since I've been there, I've really started working quite a bit on studying alpine systems. I'm working with the Niwot Ridge Long-Term Ecological Research Program to study climate change in alpine systems. But what I'm going to talk about today is kind of my bread and butter, we say, like the system I've studied the longest and that I feel the most familiar with, and that is also appropriate because it's in a Mediterranean climate. And a lot of the dynamics that I'm going to talk about today are really driven by these seasonal cycles that you're so familiar with here. So conceptually, I'm just really interested in trying to understand how species evolve to use the piece of the environment that they do. So we can think of this very generally in terms of generalists and specialists, where generalists seem to be able to have broad distribution patterns and occupy lots of different habitats. This is a picture of cheatgrass, which is a major invasive species everywhere in North America. I'm showing you the distribution here in the US and Canada. And it also seems wherever it lives, it seems to occupy almost every microhabitat. It just seems like it can do it all. On the other hand, we have species that we call specialists, right? That just occupy kind of narrow niches, we might say. This is one of the plants in the group that I study that only lives on actually islands off the coast of California in areas with high seagull densities. That's a gull right there, because they specialize on the bird guano deposits. And so in terms of ecological theory, we think about this in terms of species niches, where a niche represents the range of environments where a species can maintain positive population growth rates without immigration. And we might think of a generalist as having a big niche and a specialist as having a very narrow niche. In reality, this is very much a continuum, right? There aren't just two types of species out there. And it can also depend on which axes you're looking at. A species might be a generalist along one axis and very specialized along another. And so this example here is purple loosestrife, which is another major invasive species. And even though it has an extremely broad latitudinal range in North America, it's only found in very, very wet areas, right along ditches and things. So in some ways, it's actually a specialist, even though it's this widespread invasive species. I'm not so much interested in calling things or determining if it's a generalist or specialist as understanding what causes those patterns to evolve. What are the processes that drive a species to be able to occupy a wide range of environments? And when is it actually adaptive to really specialize on a subset of the conditions that are available? And I do this by going between thinking about this conceptual framework of niche theory and studying species distribution patterns. And we often use niches and distributions interchangeably, but I really see them as two things that feed back to influence each other. Whereas species niche is a conceptual idea about the range of environments that it could occupy, and the distribution is what it actually does occupy. And these two are connected because a species niche, where it could live, is kind of filtered by, well, what environmental conditions are actually available? How are dispersal patterns influencing if it can get to them? And how do interactions with other species influence if it actually is able to persist under those conditions? And then reciprocally, where a species actually exists in nature determines the patterns of selection that it experiences, potential for gene flow if it's in large populations or in a lot of close together populations. And as I'm going to talk a bit about today, the environments that it occupies will shape the genetic variation that is expressed, which then drives the further evolution of the ecological niche. So I'm really interested in these feedback processes where I'm ultimately trying to understand what determines where a species actually lives, but using this idea that this niche is something that is evolving and understanding how its distribution patterns influences how that niche evolves. So here's a brief outline of what I'm hoping to cover today. Like I said, I haven't talked in a while, so I'm going to keep an eye on the time and jump ahead if I need to, so I don't take more of your time than I should. I'm going to introduce you to my study system, talk a bit about some macroevolutionary patterns we've studied in both the distribution patterns and the ecological niche of the species. And then I'm going to focus on what are the microevolutionary processes that we think are driving some of those patterns or could be driving those patterns. And then I'll just a couple slides to tie it up and tell you about some things that we're working on now that I think relate to some of the projects going on here. So this is my lovely study system. It's called Goldfields, Lasthenia. It's a small genus of, relatively small genus of plants that are endemic to the California Floristic province, which is along the coast of western North America, from Baja California to just up a little bit north of California. Baja California to just outside of Northern California. So there are a bunch of plant groups that are endemic to this area. There have been a lot of radiations. There's a lot of wonderful biodiversity. And like a lot of those groups, this group is predominantly annual plants, which is one reason I love it. They have very short growth life cycles. There's one perennial in this group that occupies the coast, and there's one species that is in Chile. But otherwise, they're all found in California. About 21 taxa, 19 species, and a couple of subspecies. Oh, here's a good picture of the California Floristic province. What's wonderful about this group is I can get seeds of almost all these species in a nice road trip over a couple of weeks within California, and I can really represent the recent evolutionary history of this lineage. At the same time, they're ecologically quite diverse. So like a lot of other these endemic lineages in California, part of the reason we think they've evolved so quickly is that there's such heterogeneous environments available. And this species occupies, like I said, berg guano deposits, some of the salt flats that are the really hot, dry areas in the southern Central Valley of California. And then they're also found in these kind of coastal rolling hill areas that are more like chaparral and some serpentine sites. So even though it's a pretty young group and they're geographically very restricted, they're ecologically quite diverse, making it a good system to study niche evolution, where I can kind of more or less control for, or at least not be held back too much by geographic scope. So about 11 of the species in this group are either associated with, highly associated with, or entirely restricted to seasonally flooded wetlands called vernal pools in California. Do you have vernal? I don't know if you have them here. Seems like you should. You have the climate. I'm not sure if you have the soils, and you might call them something different. But these developed, they used to be extremely common in California, but there's very few left at this point. They occur in areas where you have open grasslands, kind of naturally rolling topography, and specifically in regions where there's this layer of soil underneath the soil surface that basically acts like almost a layer of concrete. It's just very hard and it gets saturated with water, or water cannot percolate through it. And so in the summer, vernal pools just kind of look like dry depressions in the ground. In the winter, like your winters, I guess under normal circumstances, are cool and wet, that the rain sits on top of this hard pan, and then those natural depressions become wetlands. How long they're full and how deep they get depends on how much rain there is and how it's distributed over the course of the summer, or the winter. And then as it gets hotter and drier in spring, these pools dry, and this is where you get a chance to see the spectacular botanical diversity of these habitats. So they do have about 200 species that are found only in California vernal pools. It's pretty remarkable. It's kind of a botanist dream. They're all small, so we call it belly botany because you have to get down so close. But they basically have to do most of their growth, flower, reproduce, and set seed in that short period of time from coming out of the water and being too dry to survive. And so almost all of the taxa that are in these plant species that are in these habitat types evolved from terrestrial ancestors. These aren't wetland plants. They're terrestrial plants that have moved back into the water. And because of that, they're doing something very interesting that we don't completely understand in how they manage to persist underwater. So they don't germinate in the spring. They still germinate in the fall as if they're in the terrestrial, like all the terrestrial winter annuals do. But then they just kind of manage to survive as seedlings and young plants during that flooded period. And then it's only as the water recedes that they really bolt, flower, and do everything. But they're still maintaining the same life cycle. They're just managing to survive being flooded for up to 4 months, which for a plant with a 6-month life cycle is a pretty big chunk of life to be underwater when that's not what you evolved. That's not where you evolved originally. So this group has been what we have used to study the evolution of habitat affinity and niche evolution, but it's also representative of a lot of other plant lineages in California that kind of show similar patterns, including a lot that have a lineage or two that have gone into vernal pools. This group has several, which is nice because I like vernal pools. So one of the first things that we did was we just classified their habitat affinity in kind of a coarse way using plant guides and other sources that just said this is a vernal pool species or this is a grassland species. And we broke them up into species that are designated as vernal pool, like indicator or associate species. Aquatic terrestrial, which are the ones that they're found in vernal pool grasslands, but kind of at the edges of the pools. They're not really having to get through that flooded cycle like the other ones are, but they're in otherwise very similar habitats. And then terrestrial species, which are ones that are found nowhere near vernal pools, so like true land plants. And we just plotted these on a phylogeny and evaluated the number of times vernal poolness evolved. And the most likely scenario is that there was a single transition that led to all of the vernal pool specialists, potentially a reversal, going into the pools and then back out. And then two of the species that are found in vernal pools kind of at the edges and also a variety of other habitats transitioned independently. So it looks like from this analysis, we've seen 3 kind of transitions into vernal pool habitat, but only one of those really led to the proliferation of the specialists. These aren't multiple independent transitions into vernal pools. But we also know that being here in a vernal pool is very different than being here in a vernal pool. So we went and did surveys across vernal pools that had all the different Lasthenia species in them and actually characterized their microhabitat positioning along vernal pool gradients. So even though we're talking about a pretty severe ecological gradient, these are only 30 to 40 centimeters deep. So I work in mountains and I work in very small vernal pools. So it was very easy to go through and just quantify where species occurred along those gradients all throughout California. And so then we had more of a measure of how deep they were in the pools as opposed to just in the habitat or not in the habitat. And when we did that analysis, here I'm just showing you the species that are in the pools or associated with them, because if they weren't in our surveys. And when we did an analysis of phylogenetic signal on this data, what we found was that species that are closely related are more likely to occupy similar microhabitats within vernal pools than we would expect if they were just evolving by drift. And so we see, a species, close relatives tend to be at more similar depths than we would normally expect if it wasn't something actually driving that. So this was interesting to me because I approached this question interested in all the different habitat types that these species occupy, but then we actually see some kind of resistance among relatives once they get into vernal pools in terms of their tendency to move into different microhabitats. So I wanted to actually get at that a little bit deeper and understand, well, what's constraining their distribution patterns once they get into vernal pools? Why don't we see more rapid diversification? Like you think of an islands, when species move into islands, they might rapidly diversify into a bunch of ecological niches. Why aren't we seeing that happen in this group? And what I've been talking about so far is using species distribution patterns in a phylogenetic context to try to describe how those distribution patterns have evolved, which we can sometimes is often called quantifying niche evolution using distribution patterns. What's great about this is you're actually characterizing species in their natural environments, but it doesn't actually isolate the axes of interest. And it also allows other things to be operating like competitive interactions. So if we really want to understand how these species are adapting to this semi-aquatic lifestyle, we needed to pull them out of the vernal pools and actually quantify reaction norms and tolerance curves across the gradients that we think are important in setting those distribution patterns. In vernal pools, the obvious candidate is hydrology or flooding. So we took seeds from all these different species into a growth chamber and actually manipulated the amount of flooding that they were experiencing. This is what this experiment looked like. This was at Purdue, where we had a single growth chamber. We had all of the species that are in bold, we were able to include in this experiment. So it was a good, I think it was 13 of the 20, good representation across the different habitat types. And we grew them in these bins where we just manipulated the hydro period. We simulated their natural germination conditions. They germinate kind of with the first fall rains. And so we let them all germinate under the same conditions. And then we exposed them to what we called severe drought, which was just a few milliliters of water a week, up to being completely submerged for 4 months. And these treatments span the range of the conditions we've measured in the field for these different plants, depending on if they live in a dry terrestrial environment or some of the deepest parts of vernal pools. And then at the end of our winter, we let them all dry and we just measured their reproductive output. These are annual plants, so we can actually get a measure of lifetime fitness at the end of one generation in a few months. And then I had a graduate student who was really interested in quantitative methods and developing Bayesian tools for characterizing performance curves. Characterizing performance curves is not easy. They're not linear. There were ways to characterize the curve well, but it didn't make it easy to compare among different species. Or there were ways to compare among species that didn't capture some of the nuances of how these curves could vary. So he developed a Bayesian approach. It's called performer for characterizing performance curves, where we were able to, this is an example of what a performance curve could look like. And we could actually look at the Bayesian distribution of these different characteristics of the curve, and compare them among our different species to test if they're actually different from one another. So when we just plotted those performance curves for all the different species, this is what it looked like. And what you'll notice is they all look remarkably the same in terms of where their peak is. So this group contained some species that are found in almost desert-like environments and some that live 4 months a year underwater. And yet their hydrological response curves to hydro period showed that they all prefer it to be pretty wet, but not too wet. Even vernal pool species showed reduced performance in response to flooding, which was surprising to us. When we plot this on a kind of the phylogeny, what we've done here is color-coded the green, just to help you eyeball it a bit. The green are these vernal pool terrestrial species, the ones that are kind of on the edges. The orange are the strictly terrestrial species, and the blue are the vernal pool endemics. You can again see there's a lot of variation in the curves, but they do all, some are flat, some are tall, but they are pretty much the same in terms of where the optimum conditions are. And it's not clear that some are very flood tolerant and some are very drought tolerant, relatively speaking. And in fact, if you look at those posterior distributions, so these are those Bayesian distributions, where here we're looking at where that tail hits on the dry end of the gradient. If we look at the optimum, you can see that there's no clear difference between the vernal pool species and the terrestrial species. They all have very similar optima. And in the flooding threshold, so this is where does their fitness go to 0 across the flood gradient? How much flooding can they tolerate? we actually see some variation among the species, but it's not extremely consistent in terms of the vernal pool species always being more flood tolerant than the terrestrial species. There's a lot of variation in that, although there is a tendency for this to see a tiny bit. It was not statistically significant, though. It was close, but not quite. Definitely didn't jump out at us like I expected it would. And so in these analyses of using the whole group and doing comparative analyses of their distribution patterns and their performance curves across hydrology gradients, we found that their affinity for these vernal pool habitats is relatively conserved. We saw other axes that we looked at, they had diverged quite a bit. So like the climates they occupy, their climate zones, are quite different, but their little vernal pool zone is very constrained. And their hydrological optima do not match the habitats like we thought they would. We don't see vernal pool species actually doing better in vernal pools than their terrestrial counterparts, or than they would a little bit higher and drier. So I've also wanted to investigate what are the actual drivers of these patterns. What's actually constraining those vernal pool species from becoming more flood tolerant and from diverging more, both in their habitat affinities, but also in their hydrological affinities from their terrestrial relatives. So these are questions about gene flow, selection, and genetic variation. And we have approached this by thinking about single populations. And so we're going to be focusing now on one species that kind of occupies intermediate-ish depths in deep vernal pools. It'll fill the bottom of more shallow vernal pools. And there are basically, broadly speaking, a couple of different hypotheses for what limits adaptation and responses to selection. And our approach was to think about hydrological performance curves at the level of individual genotypes now. And so if you went into this population and picked a genotype, and if we could take that genotype and grow it across that whole hydrological gradient, we could imagine it might have a response curve that represents its own performance curve, a genotype's performance curve. If we did that for a few different genotypes, we might find genetic variation in those responses. Some might do better when it's a little wet, some might do better in a little drier habitats. But if the genotype that has the intermediate performance curve that matches its distribution in the field, if that one has highest fitness, selection's going to favor that genotype and then favor that performance curve, right? So you can have selection on these performance curves, just like you can have selection on other traits. It's just a little trickier to quantify because you have to know how a single genotype does in a bunch of different environments. The alternative hypothesis is that there's just no genetic variation. We almost always find genetic variation when we look for it, so I didn't give much credence to this hypothesis. But you could imagine, for whatever reason, that if we sampled these genotypes, they would all show the same general performance curve across these gradients. In hindsight, after a phylogenetic analysis, maybe this doesn't seem like such a far-fetched idea. But when I went into this as a population biologist, I thought, oh, well, of course we'll find genetic variation. There's always genetic variation when you look for it, especially in an obligately outcrossing species, which is the one that we focused on. I showed you its distribution in the previous. It's this beautiful gold right here. It's one of these endemics, and it's in not the super deep spots, but definitely regularly flooded every year. It is an obligate outcrosser, so you have to, it can't self, and its seeds are wind and gravity dispersed. So it just, with the thought was that it's relatively locally dispersed. And we did an experiment where we generated family structured lines and planted those across the environmental gradient in order to actually characterize those genotypic response curves. So we collected seeds from a population, a very large vernal pool, and we generated paternal half-sib families So we grew a generation in the greenhouse. We did controlled crosses. The great thing about paternal half-sib families is it allows you to isolate additive genetic variance components, which is what we're really interested in terms of quantifying selection. And then we planted the offspring from those crosses across this flooding gradient. And so I've just kind of labeled it here beyond lower edge, meaning the lower edge of the population, right at its edge, right back where the population was the most, densities were highest, at the upper edge, and then beyond the upper edge. So we're kind of trying to do that whole hydrological gradient where a family can allow us to estimate a genotypic response that's due to additive genetic variance. And then we did 2 treatments, a control treatment where we just planted them and didn't do anything to the background vegetation, and then a neighbor removal treatment where we weeded out anything else that came up. Because in some of the previous projects we had done, competition often throws a wrench in things. So we wanted to make sure we accounted for that. And frankly, I actually thought competition would largely maybe or potentially be influencing patterns of selection on those response curves. So if we forget about the family structure for a minute and just look at average responses across the gradient, we saw that the control plots basically aligned with what the background vegetation was doing. They did best at the kind of center of their population. However, when we removed neighbors, we saw them shift to be more in the drier habitats. So this lined up with what I showed you for those phylogenetic analyses, where if given the chance, these species actually do best and would prefer to occupy slightly drier conditions than they actually do in the field. This isn't unusual. I mean, we often see this across environmental gradients. At the productive end, competition sets a population or species limit. And at the more abiotically stressful end, it's that abiotic stress driving that gradient. But we have also recovered that in this vernal pool habitat. Then what we did was for every genotype, we characterized its optimum niche, so which position across the vernal pool gradient did it have highest fitness. We characterized the breadth as the fitness weighted variance across that gradient. And then niche position, which is used in a lot of different ways, depending on your subfield, was just the weighted average, so the fitness weighted average of all the elevations. So if the curve is asymmetrical, then the niche position might be a bit offset from the niche optimum. So we looked at both of those. If I were to be doing this now and not in 2014, I would use the Bayesian method that we had developed, but I'm presenting this in the opposite order than we actually did it. So in those control plots, we can actually, well, in all of the plots, we can actually measure selection on these niche components. We can ask, do genotypes with a higher niche optimum, meaning higher elevation, do they have higher fitness than genotypes that don't? So actually putting the statistical tests behind that cartoon I showed you of how we would characterize selection across these gradients. And what we found was in the control plots, there was really no detectable selection for on the niche position or niche optimum, so where a genotype does the best. We did see selection for increased niche breadth, meaning genotypes that were able to kind of do relatively well across the full gradient, on average had higher fitness than those that really only showed up at a subset of the elevations. But when we took neighbors away, we started to pull out some measures of selection, some significant signatures of selection, where we saw selection for moving uphill in the niche position, selection for that kind of stabilizing selection on the niche optimum. And again, we saw the same pattern that we saw in the control plots with selection for increased niche breadth. But the big result that jumped out to us here wasn't so much these specific patterns, but just that we only saw any selection in the absence of competition, which isn't what these are experiencing in the field. They are experiencing competition all the time. And so it was surprising to see that we actually didn't measure any selection on where they occupy within the vernal pools, as we had expected. So we looked more closely at genetic variation. And in the control plots, this is now plotting every genotype's performance curve, like I had described to you in the cartoon. So we're actually looking at how every family mean did across this elevation gradient. Colors are different genotypes or families, family means. It's actually no overall effect of family, which is our test for is there genetic variation. And the effect, the families did not vary in their response to the elevation treatment. However, when we took neighbors away, we saw all kinds of genetic variations show up and a lot of differences among families in the kind of where they did best along that hydrological gradient. And if we plot this in just a slightly different way, so now I'm just plotting the family mean for those different variables, the position, the optimum, and the breadth, in the control plots and the neighbor removal plots. What you see is that in the control plots, we always have less variance in these niche components than we do when we take neighbors away. So in the presence of neighbors, we don't see much genetic variation among families in their performance across this gradient, or their, I would call it the ecological niche. But when we take neighbors away, we see lots of genetic variation expressed. So this suggests that competitors, the presence of competition, is somehow compressing the expression of genetic variation, which would then remove the ability of selection to actually drive any change in that population. This is something that has been touched on a long time ago. Fisher first developed theory behind this in terms of what is the rate of adaptation or the evolutionary potential of a population. And Fisher formalized that the genetic variation or the evolutionary potential of a population is simply the relationship between additive genetic variation in fitness divided by mean fitness. And so it's the amount of additive genetic variance there is in relative fitness in a population. And a student of mine at Purdue was very interested in understanding how evolutionary potential, which we in the previous experiment found declined in the presence of competitors. She was interested in how it would change with climate extremes, because she was interested in how plants are, especially ones that are occupying these seasonally flooded wetlands, will respond to climate change, where we're expecting more extremes between the El Nino events and the La Nina events, more prolonged droughts, things like that. So she wanted to ask the question, how does that evolutionary potential vary with different flooding regimes? And she did this again in a population biology framework, so focusing on the same species that I just talked about, Lasthenia fremontii. She collected genotypes from populations across this species range, which is really within the Central Valley of California. She generated the same kind of paternal half-fib families that I did, so she could look at additive genetic variance in fitness. And then she imposed 3 different hydrological regimes. So she exposed all of these families to 3 different flooding conditions that basically mimicked, these asterisks here identify what we were trying to mimic, a wet year or an El Nino year in a wet site, the driest year, a La Nina year in a dry site, and then intermediate conditions in an intermediate site. So trying to characterize, to some degree, the real types of extremes that these populations that this species experiences across its range and over time from year to year variation. So here's an example of her experiment. She had the 3 populations, the 3 different treatments. This is what it looked like in the greenhouse. We had lots of gnats, so we have those little sticky things to catch the gnats. And then she just, again, measured lifetime performance as survival, fecundity, and then actually seed production at the end. We were able to count all the seeds. And then this is this pattern that she found So this is again showing the family mean performance under the different treatments. And so if you were to be able to follow a line here, you would see how that family did. This is from one population, the northern population, did under those 3 different treatments. And what the main pattern that jumps out here is in all 3 populations, the families show the highest genetic variation in fitness under the driest conditions, these La Nina conditions, which she used to argue that this species has more evolutionary potential under dry years, and it could respond to selection more quickly, potentially, in dry years than in wet years, where just like with competition, these super wet conditions actually restrict the expression of genetic variation that exists among these genotypes. And so not what I expected to find, but what we have found in 2 different cases is that the evolutionary dynamics of these species within vernal pools, the evolution of their niches and the microhabitats that they occupy, isn't so much shaped by stabilizing selection, where genotypes that are in the kind of sweet spot of the niche have the highest fitness, but actually the lack of the expression of genetic variation, which is driven by ecological factors that are known to set distribution patterns across environmental gradients, like competition and abiotic stress. So I've become very interested in general in thinking about, it's very intuitive to think about how selection changes with the environment, right? Some traits are favored in one environment, others are favored in another. What I had thought less about before doing this work was how the genetic variation that's actually expressed changes from one environment to the next, and how that itself also influences the response or constrains the response to selection. So we're continuing to work on the factors that drive context-dependent expression of genetic variation in this species. So just very quickly, I did pretty well. You said 40 minutes. I'm just going to go a few more minutes. Just to wrap up, species distribution patterns in this case are mismatched with their performances across an individual axis. So even though we see these species occupying vernal pool habitats, terrestrial habitats, and in-between habitats in the field, in the greenhouse where we isolate that flooding gradient, they seem to all perform relatively similar across that gradient, showing this kind of mismatch between their niche, that conceptual framework, from their distribution pattern in the field. And that ecological factors that reduce the expression of genetic variation not as much change patterns of selection, but change the genetic variation that's expressed that can respond to selection, might actually be a really important constraint on adaptation. So something that I'm working on now, hopefully starting again soon, I haven't been able to go out to my field sites for 2 years now in this system, but is how dispersal actually evolves in association with habitat specialization. And so you might be familiar with work that has shown that species that colonize islands experience selection for local dispersal because it's not advantageous to be spewing propagules into the ocean when you're on an island habitat. I was interested in exploring that in vernal pools, which are really island archipelagos, just kind of inverse, where the wet spot's the island and the dry spot's the ocean. And one thing in this conceptual framework that I use is that dispersal comes into play on both sides of this. It both determines where species actually colonize and the environments they're exposed to, and it also can influence patterns of gene flow, which will influence niche evolution as a result. And it also occurred to me that the way I tell these species apart, for the most part, is their seeds. And so there is quite a remarkable diversity for such a young group in seed size, and then the presence or absence or the structure of this pappus, which, similar to like a dandelion seed, is supposed to facilitate wind dispersal. We see that some species have lost it entirely. Some species have it's relatively long. Others have developed strange little crowns. Seems that this is something that might be under selection in this group. And we're interested in understanding how dispersal traits evolve in response to the distribution of habitat. Are isolated habitats, for example, favoring restricted dispersal? But then how does that in turn restrict or facilitate gene flow that influences habitat specialization and those evolutionary processes? So I think when I come back to talk to Pedro's group, I'm going to talk a little bit about this dispersal stuff and get a different perspective on it. We're kind of in the midst of it, and it doesn't make much sense yet, so it's a good time to talk about it. So overall, just to remind you, I'm really interested in trying to understand how species parse up the environment, when selection favors narrowing in and just using a subset of it, and when selection favors generalist or more generalist strategies. And some of the results we've gotten from this particular system have been a bit counterintuitive, but do suggest that the expression of genetic variation is something we need to think about more in an ecological way, not just it's always there, but actually if it's there or not will really depend on the ecological context and organism experiences, which then shapes its ability to adapt to those conditions. So here's just a quick summary of people who've actually helped collect data or do analyses that I presented today. A lot of other wonderful people that I work with that I wish I could fit onto this slide. But I'd like to make sure I definitely highlight these folks. Again, thank you so much for making time for me to drop in and say hello. And I am around for a few more weeks. So if you want to drop me an email and meet for a café con leche, I can order that. I'd really enjoy any opportunity to talk more with people. Thanks.
We're also going for lunch now, so if any of you want to join, she'll be around then too. Okay, so yeah, thanks so much. Questions?
So thank you, Nancy, for the talk. It was super cool. I wanted to ask you about the second part, with the experiment you were doing, and you were seeing the niche, so the response that the plants were having to different drought or flooded moments. Mm-hmm. So you said that all of them were performing well when they were a little bit wet, but not too much wet.
Yes.
But I was wondering, how did you estimate that? Because maybe if you only do like a germination or at the initial phase of the life cycle, mm-hmm. maybe they really were very different. I mean, I guess that there is like a threshold, like for the species that are not vernal pool, mm-hmm. they have really problems to adapt to this time when they are flooded. No, I guess that must be like the main limitation. I don't know. Is that what is happening?
Yes, and so we made the assumption that they kind of all germinate under similar conditions because in terms of their hydrology, because the vernal pool species, they don't germinate after the water has flooded, they germinate before the pools are wet enough to fill with water. And so we did not look at any other axis, so just that flooding axis, really isolated that. There very much are other things that change across that gradient that could make a terrestrial species, you know, if you plant them in the field, they wouldn't germinate as well under the same conditions as a vernal pool species, for sure. But I think that those are other axes that would be really interesting. They just weren't the most obvious one to start with for us.
But your response variable was always the flower biomass for all the...
Survival and flower and, well, yes. We didn't do pollination, so we used floral biomass instead of seeds because they're outcrossers. They have to be pollinated to produce seeds. Okay. Yeah. Does that answer your question?
Yeah.
Okay.
Thank you.
Yeah.
Xavi.
Well, thank you very much for coming. Fascinating study system.
Thank you.
So I have one question about the physiology of the species, because some other species can endure some periods of time underwater, but up to 4 months, that's quite a lot.
Yeah.
Are you aware of a particularity at the physiological level allowing the plant to do that?
Yeah, we've been looking at that to some extent, and I don't claim to be a physiologist, but in terms of the vernal pool flora in general, something that's common in the species that are in these pools and that we've also seen in ours are that they have this, as seedlings, they have very small cylindrical leaves, so this like isoides strategy, which with parenchyma tissue in them. We're now looking at the roots. We don't know about the roots. What's interesting about the parenchyma is because they're completely submerged, they're not using that parenchyma to pull air down. They're completely submerged. And so the idea is that it kind of maximizes surface area to volume so that they can, gas exchange is so much slower underwater, but by having that shape, it allows them that kind of roundish shape to maximize that. And then the parenchyma tissue might actually just be a way to have cheap tissue to be able to persist. And what we have measured is that when the water dries down and they produce new leaves, those leaves don't have as much parenchyma. And so those leaves look like the terrestrial plant leaves, but the juvenile leaves, which is where their environments are different, do have this slightly rounder shape and also parenchyma tissue inside. Some of the vernal pool species, not the one we study, they've actually found CAM photosynthesis, like a desert plant strategy, but it's not about retaining water. It's just about being able to kind of maximize the efficiency of getting gas out of the water column. Yeah, they're very small plants. And I've tried to work with some physiologists about and we just haven't quite ever tweaked the equipment quite right to be able to do it, especially flooding them and not flooding them. But I think there's probably so many fascinating things these plants are doing physiologically. Something else I've noticed, since you seem interested, is they also just don't look like they grow much. It's almost like they go into a form of germinated dormancy. So it's just that ability to stop growing, I think, when they're underwater, kind of turn off a bunch of growth stuff. And their roots seem to grow quite a bit, though. So the roots are growing. I don't know if they're mining the soil for carbon or using microbes to help. I don't know what they're doing. But we see a lot more root growth and almost no above ground growth. In contrast to the ones that are in the uplands, to keep up with all of the weeds that are up there, they're constantly growing all winter. So yeah, they're turning off development in a way that I think is really potentially interesting.
Well, thanks. That's even more fascinating. So I work with the small plants, and they can do whatever.
Yeah.
So the more field work you do, the less you understand sometimes.
Exactly. Yes.
Anyway, I have another question, if I may. So, and what about the pond size and the degree of isolation among ponds? How does that affect the genetic pool within vernal ponds, and how the landscape structure affects the genetic diversity of vernal pools, which in turn can affect the expression of such genetic diversity.
Absolutely. And so getting at the population genetics of these has been something we've tried several times and run into various problems. But I think we're getting there. These are in the Asteraceae family, so they actually have quite large genomes, and we have almost no genetic tools to start with. The student Lorena, who did the work with El Niño, La Niña, she also did some RADseq, and she was interested in specifically characterizing genetic variation in chloroplast, which is only maternally inherited, and so would reflect seed movement, versus nuclear DNA, which reflects both seed and pollen. Part of the problem she had was a lot of missing data. So when we did the restriction enzymes, there were mutations in the restriction enzyme sites, and so then we didn't get any data for entire populations. But what her preliminary analysis did And that's why I don't feel confident saying, we know the answer yet, because we still got to figure that out. But her preliminary analyses on the SNPs that she was able to get across all these populations is that there's a lot of genetic differentiation when you go from kind of north to south across the species range. Within sites, there's quite a lot of gene flow, particularly by pollen, less by seed, but these are insect pollinated, so that makes sense to me. There's some genetic structure, but not as much. So it seems like the unit is probably kind of a cluster of pools at a time, which is why I feel like the archipelago is a bit, well, maybe that's even not that good of a metaphor for that reason. But they do exist in these, just the way the geology is of the area, they do kind of live in or the pools exist in kind of clumps. We have a clump of pools here, and there does seem to be genetic differences among those and not as much within. Yeah. We really want to get at that more, though, I think, especially as we're interested in understanding these dispersal traits. Like really being able to characterize gene flow across these landscapes is really important. So we're continuing. I feel like maybe by the time I retire, we will get one of these to work.
Any final thoughts, questions? All right. Well, thanks so much,
Thank
Nancy.
you so much. Yeah, it's really great to be here. Thank you.
So yeah, like I said, we're going to go to Manuela. So